Welcome to another video tutorial on electron microscope. Okay, let's get started. The next microscope is electron microscope. So yes, as you can see in the picture over here, so this is how it looks. So I think it is quite different when we compare to the other microscopes which we have studied till now. And yes, this also works with a different principle. It also have different components when we compare with other microscopes. Okay, and yes. So this microscope works on electrons instead of visible light. Okay, so now as we have studied, most of the other microscope mainly uses visible light to illuminate the object, right? So whereas here we use electrons to illuminate the object. Okay, and so apart from that, this microscope uses electromagnets as lenses. Okay, whereas in other microscopes which we have studied, so there we used transparent lenses. Okay, because visible light had to pass through these lenses, and that's the reason it was transparent. So whereas here we use electromagnets as lenses over here. And apart from that, so the entire system or the entire microscope here works in vacuum. So these are the differences here. So it works with electrons, so and electromagnets are used here, and so it works and it operates in vacuum. Okay, and yes, so this is a microscope which gives you the best images. When you compare with other microscopes which you have studied, this is the one which gives you very good quality images with high resolution and high contrast. Okay, what makes this microscope give you such good images is that the electrons which we use here, or electrons, is made up of very short wavelength. So when you compare this with the visible light, no, so the wavelength of visible light will be somewhere around 380 to 750 nanometers, whereas the wavelength of electrons are 0.1 to 0.2 nanometers. Okay, so that means yes, electrons are have very short wavelength, and because of this wavelength it gives you a very good high resolution images. Okay, and yes, so and apart from that, yes, electron microscope, no, they are quite costly. So because of their cost, high cost, no, so we don't use them in the normal laboratories. We don't use them for teaching institutions, teaching purposes, and all those things. They are only found in the specialized research institutions, universities, and they're found in advanced industrial labs like where they require high resolution imaging of nanoscale structures. Okay, and apart from the high cost, no, so they also have a very complex maintenance. They have a specialized infrastructure, and so they also need a special training to handle this microscope. And because of all these, no, we don't use them much commonly. Okay, and so for what all purposes we use this microscope is we use to study or investigate the ultrastructure of a wide range of biological and inorganic specimens. Okay, so which includes microorganisms, cells, large molecules, biopsy samples, metals, and crystals. To study all these, mostly we use this microscope. Okay, so the next is components of electron microscope. Okay, so yes, as you can see in the picture here, so this is a ray diagram of the electron microscope, and these are the following components. Okay, so now first is the source of light. Okay, so yes, the source of light here is the electron gun. So inside this electron gun there will be a tungsten filament. Okay, so there will be a tungsten filament, and when you heat that tungsten filament to a very high voltage, it starts producing the beam of electron, very fast moving beam of electrons. And this beam of electrons, okay, will be used here in this electron microscope instead of the light beam. Okay, so this is the source of light. And then apart from that, so here we use three electromagnets, coil shaped electromagnets. As you can see here, this is the first one, this is the second one, and this is the third one. So we use three electromagnets which is coil shaped, and these electromagnets here will act as a lenses. Okay, and yes, condenser lens and objective lenses are common as we see in the bright field microscope. Apart from that, we also use projector lenses over here. So all these three are coil shaped electromagnets. Okay, and so all these magnets, so they'll have different powers in them. So it mainly depends on the amount of current flowing through these coils. So based on the amount of the current flowing through these magnets, no, the power also varies. Okay, and so yes, in the normal light microscope, what happens? The lenses will be there. No? Once when the light passes through the lenses over there, the lens is used to bend or refract the light rays over there. In the similar way, so these electromagnets or these lenses over here will bend the electron beams. So their light used to get bent, whereas here the electron beams will get bent. Okay, then. Okay, so and coming in detail about the lenses, the condenser lenses focus the beam of the on the specimen. Yes, so now there are three set of lenses which is nothing but the electromagnet. So now the first set of lenses no is the condenser lens. So now the electron beam will be generated from this electron gun, so which moves through the condenser lens. Okay, and this condenser lens focuses the electron beam onto the specimen. Okay, then after passing through the specimen, the electron beam moves to the objective lens. Okay, and this objective lens is of high power and it forms the intermediate magnified image. And from the objective lenses, so the electron beam moves on to the third set of magnetic lenses, which is called as projector lens or ocular lenses, so which further produces the good magnified image. That is, as and so the electron beams are passing through these lenses, so the magnification will increase and increase, and it gives you a good magnified image in the final step. Okay, with good resolution. And apart from that, so next is sample holder. So the sample holder here, no, so here is a sample holder which will be made up of thin film of carbon. It will be made up of carbon, or it will be made up of collodion held by metal grid. Okay, so why we use carbon here, carbon films here, especially in electron microscope, is to support the biological samples. Okay, so that is especially to prevent the sample charging and improving imaging. So we use this carbon films here, and so image viewing and recording system. Yes, now the final image which will be produced, no, so they will be viewed on the fluorescent screen. Okay, why we use fluorescent screen is because here fluorescent screen is the one which converts the visible electron beam into visible light image, because see, electrons themselves are not visible to human eye. Okay, so that's why what we do is, so there will be a screen. That screen will be coated with materials like zinc oxide and phosphor materials which emits light. That is, it converts the electron beam which we cannot see through our naked eye into light which the human eye can detect. Okay, so for this conversion of electron beam into light conversion, we use this fluorescent screen. Okay, and apart from that, below the fluorescent screen there is a camera, so which is used for recording the image. Okay, so then next we'll move on to the types of electron microscope. Okay, there are mainly two types of electron microscope. So one is called as transmission electron microscope, which is TEM, and the second type of electron microscope is scanning electron microscope, which is SEM. Okay, so yes, transmission electron microscope is used to view the specimen through which electron can pass, generating a projection image. So yes, in transmission electron microscope, no, the electron beam, so they can pass through the sample, they can move through the sample, and so they generate the image. Okay, and it is similar to our conventional compound light microscope. Okay, and mainly, so this transmission electron microscope, no, so they are used to visualize the images like interiors of the cell, thin sections of the cell, the structure of protein molecules. Okay, and they're used to study the organization of the molecules in viruses and cytoskeletal filaments, and they're used to study the arrangement of protein molecules in cell membrane. So all these are the Uses of transmission electron microscope. Overall, I can tell they are used to study the ultrastructure of the cell and its components. Okay, and it can also be used to see objects as small as protein molecules, and or even you can visualize them in the nano level. Okay, so it also provides the details of internal composition of the cell or any suitable material under study. Okay, and here the thing is, based on the transmitted electrons or the produces image by detecting the primary electrons transmitted from the sample. We use primary electrons here. Fine. So this is transmission electron microscope. And then, okay, and the second type of microscope is scanning electron microscope. Okay, as the name tells here, so here the electron beam scans over the surface of the sample, whereas in transmission we saw that the electron beam passes through the sample, whereas here it just scans on the surface of the sample. And hence this particular microscope, no, they're used to produce excellent images of surface of the cells and small organisms. Okay, that is they're mainly used to study the morphology of the organisms and the cells. Okay, and so yes, here, so mainly this microscopy depends on the emission of secondary electrons. Yes, in transmission electron microscope it was primary electrons, as I told, whereas here, so we, and it depends mainly on the emission of secondary electrons. Okay, and this secondary electrons, no, so they are actually emitted from the surface of the sample only, due to the excitation by the primary electron beam. See, as you can see here, so this is a sample and the electrons, that is secondary electrons, are getting produced, and these electrons are also called as backscattered electrons. Okay, so and mainly this depends on this backscattered electrons itself. Okay, so yes, and as I told, so they are mainly used to study the surface of the cells and whole organisms that are not possible by TM, which is not possible, no? Which is they are observed here in scanning electron microscope. And so it can also be used for particle counting, size determination, and for process control. Fine, and as I told, the name here scanning electron is come because the electron beam will scan over the surface here, and these are some of the images which is produced by scanning electron microscope. Okay, so next we'll put together the applications of overall applications of electron microscope. So yes, electron microscope, as I told, it is a very good microscope which gives you the best images with good high resolution and contrast. Hence we use it in the most special laboratories, so where they used to study the ultrastructure of biological and inorganic specimens. So they also study microorganisms, biopsy samples, cells, molecules, metals, crystals, anything can be seen very clearly with the help of electron microscope. Okay, and in industries also we use electron microscope to check the quality control and failure analysis. And so today the electron microscope which we are using, no, is very much specialized. They can also produce electromicrographs by using the digital cameras. Okay, and then so yes, so we also use it in the field of microbiology. So microscope has given a very good understanding of bacteria, viruses, and other pathogens in causing diseases. And also because of it, so we have produced and we have a very good treatment for the diseases in a very effective way. Okay.